Welcome again. So today we will be now discussing about the next chapter, and that is the humoral immunity. The humoral is the term that indicates the humors, that is the liquid. So the fluid component, in the fluid component portion, that is the immunity that is circulating there, and that is the antibody dependent immunity. So this humoral immunity is different than the cell mediated immunity because it is not related to the intracellular pathogen inside the cell. This humoral immunity cannot enter inside the cell and cause or give us protection. So it is only gives the protection, that is the infection present outside the cell in the fluid compartment or say in the tissue. So this is humoral immunity that is present not inside the cell. This is from the outers extracellular pathogens, and that is actually present in the humorous environment of the body. Okay, so there is one type of immunity that is called humoral immunity, that is outside the cell, and there is another type of immunity which is related to the intracellular pathogen that is killed by the cell mediated immunity. That is a different one. Okay, so we will talk about the humoral immunity, and this humoral immunity is provided by the antibody dependent and by the antibody. So we have to understand what are the types of antibody we have, and before we have already discussed about this antibody structure. I will again discussed over here. So you can see over here, this is the Y-shape structure. So this is the Y-shape structure of your antibody. All antibody will be in Y-shape. Okay, and there will be the portions like this is the one portion. Okay, and there is the portion like this is the another portion. So the above one is the light chain and the below one is the heavy chain, and among the light chain also there is a two portion, that is like this two is the variable region of the light chain, and this two region is the constant light chain domain. So this above is the light chain, below is the heavy chain, and heavy chain is also there is a variable region of the heavy chain and constant region of the heavy chain. So we have two portion, that is the light chain and the heavy chain. In the light chain there is the variable region, that is the variable and constant region. Variable is up to this. Okay, so this portion is variable in both case. Okay, this portion is variable. Here also this portion is variable, this portion is variable, the below one is the constant. You have to understand in a such a way that the above one, this one is actually this one is the light chain one, and this one is the heavy chain one. Okay, the heavy light chain also have the variable region and constant region. Heavy chain also has the variable and constant region. And one more thing, important thing is that this portion, this portion that is formed by the both variable region of the light chain and heavy chain is the place where antigen binds. This is called antigen binding site. So that you have to understand. This is the place where antigen gets bind. This is the place. So if you understand, and this is the place where the complement get activated, opsonization occur, or other things occur, but the antigen is binding in this portion. This is the portion where antigen binding, it is variable, so it can change its structure by the variable combination and bind to the variety of antigen that is present in the environment that will attack your body. So let's now move towards our chapter. So we are talking about the humoral immunity. The main objective is explain the information related to the primary humoral response and answer the questions about antibody of secondary immune response. So primary humoral response, the first isotype of immunoglobulin that can be produced by the B cell with or without activation of the T cell is IgM. So IgM is a protein, IgM is a immunoglobulin, IgM is the antibody that will formed inside your body after the activation of the B cell that will convert into the plasma cell and secretes this IgM antibody. This is actually IgM is incorporated as a receptor in case of the IgM and IgD. So IgM and IgD are the receptor of this B cell, and this B cell will convert into plasma cell. So the first thing that will be released is IgM, that you have to understand. The first isotype of immunoglobulin that can be produced by the B cell with or without help of the T cell is IgM. Now you have to also understand why this T cell is not required to convert into the plasma cell, because previously we have discussed that B cell can be activated in the two way. One, it will directly activated by thymus independent pathway, thymus independent antigen, that the antigen that is polysaccharide or lipid or lipopolysaccharide, that will not be presented by the T cell. This is not thymus dependent, that they are thymus independent. So thymus independent, what will happen? Thymus independent antigen will be processed and presented by B cell directly. So the B cell will be activated and they will release the IgM in the environment, and they will activate the complement and then kills the pathogen. So this will be weaker immunity, less long lasting and weaker immunity, although there will be the secretion of IgM. So if it is processed and presented by, if it is a protein in nature, the component, the antigen, that will be processed and presented by MSC class 2, then to the T cell, that will the helper T cell, and that will come by the B cell, and then B cell will activate it, and this will release the initially they will release this IgM, and then again there there are the two wave. If you remember, there are the two wave of antibody secretion, and that is very important to understand. So there is actually primary wave of activated B lymphocyte is comprised of strictly secreting IgM. So there is in the two phase the antibody is released, that is the primary wave of activated B lymphocyte is comprised of the strictly IgM secreting plasma cell, which leave the secondary lymphoid tissue shortly after being activated. So as soon as activated, this will leave the lymphoid tissue, secondary lymphoid tissue, and secrete this IgM. This is the primary wave. There is another wave. After a few times, the second wave of activated B lymphocyte remains within the secondary lymphoid tissue, undergoing the clonal expansion, producing into the germinal center. So in the germinal center, there is still this activated B lymphocyte, that is the plasma cell is there, and that is going under the clonal expansion, and this will undergo the affinity maturation and isotype switching. So the secondary wave will again there will be the affinity maturation and there will be the isotype switching. So now the release antibody from this isotype switching will be the immunoglobulin G or immunoglobulin A or immunoglobulin E, depending upon the signal that will send by the helper T cell. So you have to understand the antibody formed, it is not the IgM that will be converted into IgG. It is actually the second set of the lymphocyte that is activated, that is the second set of the plasma cell that is remain in the germinal center, that will provide, that will secrete the antibody that will undergo class switching, and that will produce the immunoglobulin G, A or E. That's depend upon the signal that will send by the T cell or helper T cell. So hope you have understood about this, and this was the class switching mechanism. You can understand, we have discussed about the VDG classification, and if there is mu, then the first concept in the first sequence there is the immunoglobulin M. If it comes to the D, then it will be immunoglobulin D. If it is the gamma, then immunoglobulin G. If it is alpha, immunoglobulin A, and if it is the epsilon, the immunoglobulin E. So during the splicing, during the looping out, which comes forward? Initially in the chain, the first is immunoglobulin mu. So there will be always immunoglobulin M will form. After that, there will be when there will be the class switching, then there will be looping out, and which will come in forward. Suppose immunoglobulin, that is the gamma 1 has come, so there will be the immunoglobulin G1 will form. So this is the way of immunoglobulin heavy chain switching. So this is called class switching. So that's occur only in the second set of activated B lymphocyte. Primary, there will be always immunoglobulin M. This is because the coding of the constant domain of the heavy chain of immunoglobulin M are the first sequences downstream from the coding for the iodotype of the molecule, which we have seen. This is the first sequence that has been coded, that is the mu, and that is the immunoglobulin M will always come first. In the first response, there will be always immunoglobulin M. So we have understood the hemoglobin M on the surface of the B cell is monomer, but secreted from this molecule is pentamer, held together an extremely compact form by the J synthesized by the cell. So the B cell, what is happening? There will be the monomer, the one component will be there. So this is, suppose this is the monomer, one unit, and that is only present as a receptor on the B cell, but while secreting they are joined together by this J chain. This is J joining J chain, and then form the pentamer. So the 5 single unit is attached to one another by the help of the J chain when they are secreted, when they are released. This is the structure of the immunoglobulin M. So on the surface of the B cell, it is only in single unit, that is monomer, but while secretion, they will be in the pentamer, that is held together by the J chain synthesized by the cell. So you have to understand about this pentamer, why it is called pentamer, and one antibody has 2 binding sites, so the valency will be 10, which we will discuss over here. So there will be the valency of the 10, and one more thing, their affinity is decreased, means their ability to bind to the antigen is less, but since they are pentamer, they can bind 10 times or say 5 times more than the normal immunoglobulin like immunoglobulin G, and this has the higher avidity. So we'll talk about the affinity and avidity. The design of the IgM pentamer maximizes its critical to the body early during the antigen challenge because of the multimeric structure, that is the 5 of the Y-shape monomers join as into one unit. Plasma has 5 times the capacity for binding antigen epitopes, that it has more antigen binding epitopes. The valency of the molecule is therefore 10. In other words, 10 identical epitopes can be simultaneously bound as compared to the other monomeric unit that has the 2 binding site. The design of the IgM most effective immunoglobulin isotype sponging free antigen of the tissue and provides critical as the humoral response evolves in trapping the antigen, so it can be presented into the lymphocyte. Now we need to understand about this affinity and avidity. Okay, so talking about the affinity means the capacity of binding this variable region. So the capacity of binding this variable region towards the antigen, the attraction, the law of attraction, the force of attraction is more in case of immunoglobulin G, that is binding towards the antigen is more in immunoglobulin G in comparison to the immunoglobulin M. So immunoglobulin M has less affinity, but the problem is that this immunoglobulin G has only 2 binding site, so there is avidity is only 2, but in case of this immunoglobulin M, they has the actually 10 binding site. So the maximum number they can in the immunoglobulin M, there more antigen can bind, and then more capability, the total capability of binding antigen is called avidity. So the avidity is more in immunoglobulin M, but the affinity is more in immunoglobulin G, that you have to understand. So let's be clear, the affinity is more of immunoglobulin G, avidity, that is total binding capacity, is more in immunoglobulin M, that you have to understand. The multimeric structure of IgM also makes it most effective antibody against activating the complement. So it can activate the complement, a set of protein, you know about the complement, but this is not responsible for like ADCC opsonization or other thing. So like immunoglobulin G, they can activate complement, they can do ADCC, they can opsonize, but immunoglobulin M, if you talk about the function, that has only one function, that is activate the complement. Okay, there is one disease called Xlink hyper IgM syndrome. The name itself is saying it is the Xlink hyper IgM syndrome. So there is some disease that is related to Xlink, and that in that case there is high hemoglobin M inside your body due to any reason, and we need to find out the cause why this IgM is high inside your plasma or in your body, but other is not there, because it is characterized by the deficiency of immunoglobulin G, A and E. So what is happening? Immunoglobulin is M is present, but other immunoglobulin is not present because there is no class switching occur. And what could class switching? What is the major role that class switching occurs? Because of the signals that has been sent by the T cell, and the most important signal was the co-stimulatory signal of CD40 and 40 ligand. So anyway, if the CD40 ligand is not there, then class switching will never occur. So there will be only immunoglobulin M inside the body, and other immunoglobulin will not be formed. So this is the characterized by deficiency of immunoglobulin G, A and E, and elevated level of immunoglobulin M. This can reach to up to 2000 milligram per dl. Normally we know it is 245 to, so it is below 250. You can understand it is mostly commonly inherited as X-linked recessive disorder, but some forms seen to be acquired and can be seen in both sexes, that you have to say. Peripheral blood of the patient has high number of IgM secreting plasma cell as well as autoantibodies to neutrophil, platelet and red blood cell. Patient failed to make the germinal center during the humoral immune response, so there is no class switching. Children will this condition suffer from the recurrent respiratory tract infection. Why respiratory tract infection? Because respiratory tract protection is done by the immunoglobulin A, and since there is no immunoglobulin A, now patient has only immunoglobulin M that can only activate the complement but doesn't protect the mucosa of the respiratory tract. Then patient will suffer from the disease called recurrent respiratory tract infection and pneumocystis jirovecii. Why this has occurred? This defect of syndrome is seen in the gene encoding the CD40 ligand, which maps the X chromosome. Therefore, TS cell of this patient will fail to express the functional CD40 ligand on the membrane, failing to give the co-stimulatory signal needed for the B cell response to T dependent antigen. As a result, only IgM antibodies are produced. The B cell responds to T independent antigen is unaffected. So what is happening? The T cell that will give a co-stimulatory signal and activate the B cell and convert it into the plasma cell, that process is not there. So immunoglobulin M is forming, but immunoglobulin class switching is not occurring. So there will be the high immunoglobulin level in the body, but other immunoglobulin deficiency, and this will mainly affect the T cell dependent plasma cell activation, that is thymus dependent. So there is no thymus dependent activation, so there is no formation of other classes of immunoglobulin G, A and E, and patient will develop this hyperlinked immunoglobulin M syndrome, that is related to due to the absence of this CD40 ligand on the T cell that fail to give the co-stimulatory signal to the B cell, and because of that thymus dependent antigen will be not able to class switch, and therefore there will be only IgM in the body. Now talking about the other class switching. So there will be the class switching. So we have understood this is the condition where class switching not occur. That is a disease called hyperimmunoglobulin M syndrome. This is the Xlink hyper IgM syndrome. Now we will talk about the normal condition. Normally what happen? There will be the thymus dependent antigen protein in nature that will be processed and presented to the B cell and converted into the plasma cell. Now there will be class switching to the immunoglobulin G, immunoglobulin E and immunoglobulin A, depending upon the signal that will be sent by the T cell. So for switching to the immunoglobulin G, there is the requirement of this interferon gamma produced by the Th1 cell. So the predominant isotype of immunoglobulin that begins to produce after IgM during the primary immune response is IgG. IgG is the monomer molecule with the heavy chain, gamma heavy chain, and new set of the effector function. A majority of IgG is produced in response to the interferon gamma, that is INF gamma produced by the Th1 cell. So after B cell, there will be a T cell. B cell will bind to the T cell. There will be the presentation of your antigen. Then there will be the CD40 and CD40 ligand co-stimulatory signal. Then that B cell will be converted into plasma cell. And after that, if interferon gamma is coming, then that will class switch to immunoglobulin G. If interleukin 5 will come, then that will class switch to interleukin 5. Then it will switch to immunoglobulin A. If it is interleukin 4, then it will switch to immunoglobulin E, and this is EVER, ever, A always, ALYS always, A 3 4 5. So you have to remember this is 5 for always, 4 that is E for ever, and if it is interferon gamma, INF gamma, then it will release to immunoglobulin G. So this is very important to remember. You can remember by any means, although letter 5 is E, but it is not interleukin 5 that is activating it. Interleukin 5 is responsible for immunoglobulin A, interleukin 4 is responsible for that is immunoglobulin E, and interferon gamma is responsible for immunoglobulin G. So what is signals is produced? According to that, the class switching will occur. This totally depend upon the what? Depend upon the this interleukins that is been secreted from the T cell. So the class switching, the predominant isotype we have discussed, the majority of immunoglobulin is produced in response to the interferon gamma. So this interferon gamma produce the Th1 cell. Immunoglobulin G exists in 4 isotype, that is immunoglobulin G1, G2, G3 and G4. Each